So, good evening, and you're looking at Bio 211 Test 2 review. And what I've decided to do is put together probably 2 or 3 YouTube videos here about the review material for chapters 4, 5, and 12. Chapter 4 being connective tissue, chapter 5 being skin or integumentary system, and 12 being the introduction to the neuron, the single cell, and the supporting cells required for that, which are glial cells. What we'll do is we'll work our way through those first 11 review questions that I gave you guys for the review for test number 2. You'll find those on say Blackboard if you haven't been able to look at them yet. I recommend you do the reading first before you look at them. And then listen to what I've gone through here to help you understand what I'm asking you to learn. The idea, of course, is you come to class a little more prepared for an exam on Wednesday. Okay? So let's get to work. The first question in the review packet was, what is a tissue and what are the 4 major types of tissue that are developed inside Chapter 4? Can you describe in detail how liquids such as blood and lymph to bones can be grouped together as connective tissues? And the way I tend to think about this, of course, is that drawing that I put in on all of the sort of explanations that I've put so far. But remember that all major tissues are derived from endoderm, endo, ecto, and mesoderm. Okay. And that when we're thinking about how we group them, remember what I've done is I have pretty much grouped them such that what we do is, I'm not really good at this yet, so we're just going to bear with me, okay? We break them up into these groups, right, as these 4 types here, all right? And because they're all tissues, I'm just going to put the T here, but neuronal over here, muscular over there. Let's go connective tissue over here, and let's go epithelia over here. And you should be able to, at least in the chapter 4 sense, what gives rise to endoderm, what gives rise to ectoderm, or what arose from each of these. So like the nervous system primarily, as far as chapter 4 and chapter 3 are concerned, nervous system arises from ectoderm. Mesoderm gives rise to both muscular tissue and connective tissue, and endoderm gives rise to epithelia. The farther you go along, you realize that there's much more complexity than this, but for this exam, that's what you need to make sure you understand. Okay? Let's do this. Get rid of all of that. The next is, once you've been able to figure out how to group them together, what is significant about them? Remember, what I've done so much so far for you guys has been so that you realize that when we break apart the tissue types themselves, that they're grouped. Okay? And what do I mean by that? If we take it if it's just muscular tissue, muscular tissue, right? Sorry about that. Muscular tissue, right? You're automatically thinking, let's fix that. You're automatically thinking skeletal, cardiac, and smooth. And you'll notice that I gave you structural characteristics. Structural characteristics. Carac. Okay? And that's probably what you want to make sure you understand for this exam. You know, how is skeletal muscle tissue different than cardiac and smooth? Well, obviously, where do you find skeletal muscle tissue versus smooth muscle? You know, the smooth muscle is found in viscera. Why is that significant? It's because it's regulated autonomically, meaning we don't voluntarily control it. Likewise, if we think about epithelia, right, we group that, you can see that we use epithelia for cutaneous or external, and internal structures. And the way we group those together is external require glands that create oils that support their existence. Whereas inside of our body, we have cells that secrete materials that draw water to them. So you would think of lamina propria here. So cells that are secreting materials that draw water to ensure that the tissue never dries out. You think about, say, the gastrointestinal tract, or if you think even the lamina propria inside your throat. Okay? I'm going to draw my cartoon again in a second, but notice what I'm doing here. I am literally grouping these things together for you so you have an understanding. Okay? Connective tissue, now this is probably the most difficult, right? Where you actually go from liquid, QUID, to solid. And each of these has a characteristic, right? And what do I mean by that? So liquids, we can be talking about lymph, LYMPH, or for that matter, cerebral spinal fluid, the CSF. Okay? How are these made? What are their characteristics? That's what you want to pay attention to. Okay? And then when you get to solid, what are the patterns of bone growth? So remember, there are two types of bone growth. Right? There's compact bone growth and there's trabecular bone growth, or spongy bone and compact bone. And, there's a pattern here that you want to make sure you're paying attention to those little details. I'm not sure there's anything that's easy about these, but in terms of characterizations, nervous system is, of course, the easiest. You either have a neuron, N-E-U-R-O-N, which is an excitable cell. You'll spend all of chapter 12 figuring that out, and by now you have a pretty good idea about it. But also inside the nervous system, we have non-excitable glial cells. Okay? And depending on where you are and your understanding of things, these are in many ways just as important, if not more important, to how the entirety of the nervous system works. So let me draw this one more time just to make sure you get an idea of what I'm trying to make sure you can do on a test, right? So this sort of idea. Let's go, this is muscle here, right? And remember, all I did was something like this, where you have skeletal muscle, you actually have smooth muscle, and then you have cardiac muscle. And what are the characteristics of each of these? You should be able to do that for all of the actual, so let's go neuron here, and let's go glial cell down here. Glia. Okay? Each of these should be sort of almost, I can't say second nature to you at this point in time, but that's what we're thinking about when we're looking at question 1. Okay, question number 2. What about epithelia? How do you hold on to them? All this sort of stuff. What's found underneath them? In fact, is it empty? What are the types of materials? I'm asking multiple questions here. And a way to think about the answer for this would be something like this. Here is one cuboidal epithelia, and it's sitting on top of a basement membrane, right? And inside of this basement membrane, we have vasculature, all right? And that vasculature uses diffusion to keep that epithelial cell alive, and this is its free apical surface. How does it hold on to this cell that's adjacent to it? What were the tools? How do these things actually hold on to one another? You need to know those. How does it hold on to the basement membrane? You need to know those. Okay? That's what I'm talking about. You'll notice that there's a series of rules here. This is the lateral side here. This is the basal side here, and this is a free apical surface. But if I put this inside of a tube, and I lined it with endothelium, or epithelia that is inside of a tube, and notice I changed its name to endothelium, now we actually have a free luminal surface here with a free apical surface, and you still have a basement membrane that's surrounding that. That's how we build things like lymph vessels, arteries, and veins. Okay, what's inside this space down here, inside the connective tissue part? It's not empty. We know that. We know the major cell down here is the fibroblast. Fibroblast. And that fibroblast is making materials, and those materials are glycosaminoglycans. And what are the different types of glycosaminoglycans? What is chondroitin sulfate? What is hyaluronic acid? I'll put HA here. Hyaluronic acid for that. What are they doing? Why are they functioning where they're functioning? Okay? So that's what I'm thinking about when I'm looking at this question. But notice all the details here. Can you define all of these structures for me? Cool. Next question. Man, we might end up making many more than 3 slides here. Let's go to number 4. Okay. Why would both muscular and neuronal tissues be grouped together? Okay, what are the reasons that they are different types of tissues? So again, I'm going to go to that quick little drawing. So we have tissues here, right? And remember, we went like this, and we went like this. How many times have I drawn this for you now? You're not going to see it on a test. What I'm doing is I'm teaching you how to think about the problem. I'm hoping that's what I'm teaching you, right? So we'll put epithelia over here, connective tissue there, muscular tissue here, and nervous tissue here. And the reason why these guys are grouped together is, of course, they're excitable, right? Sorry about this. There's a lag period inside the pen, and we're seeing it on the screen. Okay. Great. Okay, so they're grouped together because they're excitable. Yay. Who cares? Well, you care. We spend all of chapter 12 learning how we make a graded potential become an action potential that then releases neurotransmitter into a cleft that will then cause another graded potential to become an action potential if the stimulus is excitatory to cause now the release of another neurotransmitter. Make sure that's what you're getting when you're studying this nervous system stuff. Whereas muscular system, we haven't gotten to yet, right? But you know what I taught you about this is regardless of the muscular system type, it is a contractile structure, which means we shorten the distance between 2 points, pulling A towards B. Okay? Cool. That's the reasons why they're different types of tissues. Mother Nature needed to create something that could contract and move materials towards one another, or she could send electrical signals in the shape, literally, of up here, a graded potential that is excitatory. So that'd be an excitatory graded potential versus an inhibitory. So that's a depolarizing graded potential. And then we have our action potential, right? That's traveling along the axon. And then we actually release neurotransmitters that will then communicate to the next adjacent cells. That's the pattern. Next. In chapter 4, hopefully you see that what it did is it drove home the idea that tissue is required. How is what you learned from chapter 4 different from chapter 5 and chapter 12? Okay, so here's where chapter 5 is really what I'm asking you about right now, right? Because what I'm saying is, do you realize that chapter 5 is an entire system? SYSTEM. What the hell is going on here? Okay, let's go like this. Let's erase all of that. I'm going to go like this. Let's get out of there. Let's go like that. I'm going to go like this. I'm going to start answering this question by thinking about chapter 5. Whoops. Drop down here. Chapter. PTER 5 is a system. SYSTEM. Do you understand how it's functioning? So you actually have all tissues in it to function. Right? Whoops. Or spelling can get you in trouble. Okay? Whereas chapter 4 is all tissues that are not assembled They're not assembled. You have to assemble them. You have to go look at examples of each example. My fingers can't keep up with my brain, sorry. Examples of each. Nudge, nudge, hint, hint, wink, wink. Make sure you understand that we've given you examples of each of these inside the textbook, and you should be aware of what they are. Okay? Whereas chapter 12 is how does a single neuron, N-E-U-R-O-N, work? That's all we did. We discussed glial cells? Sure. Glia. And remember what we did was we built a blood-brain barrier, but I didn't stress that to you. You need to know what the different glial cells are, what their responsibilities are, of course. But think about what most of chapter 12 was about. Most of chapter 12 was literally about, can you draw and understand what's happening, in this case, in a multipolar neuron, right? Think about the fact that when we're describing how they're different from one another, everything inside chapter 5 is what does a keratinocyte actually do? What do cells that make melanin do? Okay? How do we actually have nervous system structures that innervate the integumentary system? Whereas we don't talk about that here. We talk about what are epithelial cells. Chapter 5 is what happens when we build all of these. We layer all these epithelial cells in a stratified manner. What's significant about that? Why do we have two types of these? And then what's that basement membrane thing again? You know, why doesn't the epithelial cells, or why don't they function unless you actually have this in place? I don't know what's going on with this pen. But let's get back to this chapter 12 piece down here, because I'm going to say it again and again and again. We're about to end this one right here, but what's happening in this part of the neuron? What's happening in this part of the neuron? And what's happening in this part of the neuron? So when we pick up on the next question, what I'll do is I'll go through, hopefully a little bit more detail. Okay?